नवयुवक शिक्षण प्रसारक मंडळ चापोली संजीवनी महाविद्यालय चापोली welcome you in e-learning platform prepared by myself Dr. Shubhangi Chavan, Assistant Professor and Head Department of Botany. Now, Swami Ramanand Tirth Marathwada University Nanded Semester Pattern Curriculum under Choice Based Credit System CBCS for undergraduate program, Faculty of Science and Technology, subject Botany, year BSc second year and third year, paper Plant Physiology. Now, in previous lecture we learned minor elements. Now, in today period, let us learn translocation of solutes. First of all, sour and sink relationship. All green leaves photosynthesizing producing complex food material are considered as source while all other organs of the plant utilizing carbohydrates are regarded as sinks. The carbohydrates is transported from source to the sink, appear leaves on the plant, exports principally to the shoot apex, while the lower leaves to the roots and middle leaves to the both roots and stem. The translocation of sugars from source of sink takes place through serve the tube to the both sides of stem. The leaves on one side may be more efficient at exporting carbohydrates or other assimilates to sink on the same side. Now, however, in many species of plant, there is more cross in linking the phloem tubes specially or nodes, and these elements on one sided transported of assimilated during the translocation from source to sink. The sugar synthesized in leaf cells move simplistically to the serve tubes. During this, there is rise in simplistic concentration of sugar above the apoplasts. Now, the sugar at the sink end is absorbed and either actively converted into constituents or two other carbohydrates which have very little effect on the osmotic potential and hydrostatic pressure of the phloem. The loading of the sugar in the phloem at the source end and their removal or conversion of sink end produce on osmotic potential or hydrostatic pressure gradient. This gradient causes the movement of water and sugars from the source to sink. Now, with the increased photosynthetic activity in the source cells may increase the rate of translocation to sink cells. However, sink cell must be able to utilize sugar efficiently so that the deficiency in hydrostatic pressure is maintained. If they are unable to utilize the increased production, there will be steadily blood of sugar in the system causing a feedback inhibition resulting in reduced photosynthesis. This sink capacity will determine the rate of photosynthesis in source cells. Now, next point, translocation of solutes. The solutes are broadly classified into two groups. First of all, inorganic solutes. This includes mineral or inorganic salts. And second, organic solutes, non-electrolytes. These include synthesized food such as sugar, proteins. First of all, translocation of organic solutes. Now, organic solutes are translocated in the phloem. Carbohydrates account for more than 90% of the substance translocated in the phloem source in the most abundant carbohydrates, sugar, alcohols, maltose and sorbitol have been detected in the phloem. Exudate of some species, nitrogenaceous compounds, mainly amino acids, are translocated out of sentient leaves and flowers and transported to the younger parts of the plant. In addition of upwards and downwards translocation of organic solutes, phloem tissue are also responsible for the radial and tangential movement of the materials. Now, next, mechanism of organic solute translocation. Several theories have been put forth of the explain the phloem translocation of the solutes such as mass flow hypothesis and protoplasmic staining theory have been considered as significant. Now, diagram first. This diagram shows mass flow through phloem. It indicates the flow of solutes from root to leaf and leaf to root through xylem and phloem. Now, next, diagram second. Munch osmotic model to explain the mass flow hypothesis. And last, diagram third. Diagrammatic representation of protoplasmic streams of hypothesis. During diffusion of dark and clear circle occurs through the cell plate in response to their own concentrating gradients. These two kinds of particles will move simultaneously in opposite direction. The diagrammatic representation of mass hypothesis. Now, next, mass flow hypothesis or pressure flow hypothesis, Munch hypothesis. This theory was developed by E. Munch 1927 and was supported by Kraft 1961 and Issue 1966. This theory is based on the assumption of extents of the turgor pressure gradient between the supply tissue, mainly mesophyll cells in the leaves, and receiving tissue from the plant organs such as the root. Now, according to much of osmometers corresponding to the seats of assimilation, mesophyll cells of leaves, and B to the seats of storage, such the tree trunk underground storage organs, the glass tube T transport the serve tube phloem and through containing water corresponding to xylem elements. Now, main objection of Munch theory. First of all, mass flow hypothesis account only for unidirectional flow of metabolic, but biodirectional flow is the common occurrence in plants. Now, second, mass flow hypothesis suggests that the translocation of metabolites in the passive pressure, but translocation of solutes requires an expenditure of energy on the part of the series tube elements, and Sohn's 1959 has further demonstrated that ATP accelerates translocation rate from mesophyll cells to phloem cells. Now, next, protoplasmic staining hypothesis. This theory was proposed by Hoge D. Various 1885 and was supported by Curtis 1935, Thanian 1962 and Canny 1962. This theory assumes that the movement of the solute particles is caused by the combination of diffusion and cytoplasmic streaming. It is presumed that these solutes particles diffuse from one serve tube to another serve tubes across the intervariance cross wall pore through cytoplasmic strains. Now, the translocation of organic solutes is bidirectional in the phloem and unidirectional upwards in the xylem.